Welcome! If you're gearing up for your Cambridge IGCSE Biology exam, you know there are some topics you just have to nail. Well, today, we're diving deep into one of those fundamentals, the structure of a bacterial cell. So let's get right into it.
But hang on, before we put the cell under the microscope, let's just zoom out for a second. Why is it so important to understand a simple bacterium? Well, because biology isn't just about memorizing facts for a test, right? It's about getting the building blocks of life itself. It's stuff that prepares you for university, for amazing careers, and honestly, just helps you make sense of the world.
And hey, don't just take my word for it. This kind of prep is recognized all over the world. I mean, look at this quote. Top universities really value this curriculum because it teaches you to think, not just to remember. What you're about to learn today is a perfect example of that powerful foundational knowledge.
And here's how you're doing that. When you learn to describe a cell, you're not just listing parts, you're practicing precision. When you figure out what those parts do, you're problem solving. It's all about applying that scientific method, you see? These are the skills that turn biology from just another subject into a powerful tool you can use anywhere.
So, with that bigger picture in mind, let's get down to business. Time for our specific mission in this explainer. We are going to decode one very specific but super important part of your syllabus.
And here it is, straight from the source. Our focus today is section 2.1.2, describe the structure of a bacterial cell. The syllabus lays out exactly what you need to know, and that's what we're going to cover. Every single part, no more, no less, just the good stuff.
And here's our roadmap for today. First, we'll get into the bacterium's basic anatomy. Then, we'll check out its genetic code. After that, and this is the cool part, we'll connect those tiny parts to why bacteria are such a big deal in the real world. And finally, we'll wrap it all up with your key takeaway for the exam. Sound good? Let's do it.
Okay, let's start with the anatomy. To make this super clear, we're going to work our way from the very outside layer and move inwards. It's the most logical way to build our understanding.
First up, you've got the cell wall. I want you to think of this as the bacterium's suit of armor. Seriously, it's this tough, rigid outer layer that gives the cell its shape and, more importantly, protects it from all the crazy stuff in the outside world. It's all about that crucial structural support.
Now, tucked right inside that cell wall is the cell membrane. So if the wall is the armor, think of the membrane as the club's bouncer or the security guard at the gate. Its job is incredibly important. It controls exactly what gets to come into the cell and what has to leave.
Okay, let's pause here, because it's super common for students to get these two mixed up. So let's be crystal clear. The cell wall, that's on the outside, it's for structure and it's fully permeable, kind of like a net. Lots of things can just drift through. But the cell membrane, the inner layer, that's all about control. It's selectively permeable. It's picky. It chooses what gets in and what gets out. So remember, one is for structure, the other is for control. Got it?
All right, we made it past security. We're now inside the cell, floating around in the cytoplasm. This is basically the jelly-like goo that fills the whole cell. You can think of it as the factory floor. This is where all the action happens, where most of the cell's vital chemical reactions are going down.
And scattered all over that busy factory floor are the ribosomes. Now, they're tiny little structures, but don't let their size fool you. Their job is huge. Ribosomes are the cell's protein-making machines. They carry out protein synthesis, building every single protein the bacterium needs to function and survive.
Okay, so we've covered the cell's basic structure and machinery. Now let's talk about the main office, the control center, the genetic code that holds all the instructions for everything the cell does.
In a bacterium, the main set of blueprints is the single loop of circular DNA. Now, for your exam, two things about this are absolutely key. First, it's a circle, not a straight line like our chromosomes. And second, it just floats freely in the cytoplasm. There's no nucleus to keep it safe. But wait, there's more. Bacteria also have these really cool extra bits of genetic code called plasmids. These are small, separate little rings of DNA. You can think of them like extra apps on a smartphone or special toolkits. They aren't essential for the bacterium's everyday survival, but man, they can carry some super useful genes, like the instructions for how to resist antibiotics. And as we're about to see, this is a huge deal.
So now we know the 6 key parts, but why does a tiny little thing like a plasmid even matter in the grand scheme of things? Well, this is where your IGCSE syllabus does something really cool. It connects the dots from these tiny microscopic structures to massive, world-changing industries.
So let me just ask you directly, what do you think? Looking at that definition, what is the big deal with plasmids? What makes them so special?
Well, check this out. The answer is literally right here, in another part of your syllabus. Section 21.1 spells it out. One of the main reasons bacteria are so incredibly useful in things like biotechnology is specifically because of the presence of plasmids. See, it's all connected.
Here's how it works. Scientists can use these plasmids like tiny little delivery trucks. They can take a human gene, like the one for making insulin, and actually splice it into a plasmid. Then they pop that modified, or recombinant plasmid back into a bacterium. And since bacteria reproduce like crazy, pretty soon you have billions of them, all pumping out human insulin for us. It's absolutely revolutionary stuff.
And that brings us to our final takeaway. We've gone through the whole structure, we've seen a mind-blowing application. Now let's just lock in exactly what you need to have down cold for your exam.
So, what's the bottom line? For your exam, you need to know these 6 key parts of a bacterial cell. Let's run through them one last time. The cell wall, the cell membrane, the cytoplasm, the ribosomes, the main circular DNA, and of course, those game-changing plasmids. That's your checklist.
So we'll end on this thought. This tiny organism we just explored, on one hand, it can be a deadly pathogen. On the other, thanks to those plasmids, it can be a life-saving factory. It really makes you wonder, with everything we're learning, what are we going to discover it can do next? Thanks for tuning in.